I'm really delighted to be here today to talk to you about my research on heart development and evolution, which hopefully will be projected on the screen. Okay. And I'm going to start by giving you a broad framework about our research, which has to do with the conversations, the cellular conversations that are going on in developing embryos that help to establish the identity of different cells. I'm then going to give you a brief introduction to the organism we use to study these questions, Ciona intestinalis, and then I'm going to talk to you about what lies beneath these hidden conversations and how we listen in as researchers to these conversations between cells as they establish the identity of the heart and other organs. So I'm going to actually begin this, introduce you to this topic the way that I introduced my freshman students to the topic in Intro Bio, which is to have them watch two time-lapse videos, one, the construction of an embryo, and another, the construction of a building. And have you just watch those videos and discuss with your neighbors for a minute what you think are some of the fundamental differences between these two modes of construction. So I'm actually going to open this up to you and really just watch the videos and talk to the people next to you and try to come up with some ideas about what's fundamentally different about these two processes. Okay, so I'm gonna your minute are a little short. I give the students a lot longer to think about this. They actually see these videos the night before the first lecture and have a lot of time to develop their ideas about this. And instead of soliciting your responses like I would do with them, I'm going to actually show you some of their responses from this year, which are really impressive. And I'll just read two of them. One is the fundamental difference was agency. That is, the embryo is building itself. Another is the construction of the embryo begins from a single unit. And you can see some other really interesting and insightful responses that I got from the students. And this is why I love teaching Swarthmore students. But their answers really dive right into the central mystery of embryonic development. And that is, how is it that a single cell is able to self-generate a much more complex structure than is a multicellular organism? So, one of the key questions within that central mystery is how are cells creating a pattern? In order for cells to create a pattern of cell identities, they have to somehow be coordinating those identities with their neighboring cells. In order to coordinate those cell fate decisions, cells have to communicate with each other. And this cellular communication is absolutely fundamental to development, and you can think of this as this intracellular conversation between the cells. And you have to think about those conversations as occurring in a very dynamic environment. As these cells are dividing and moving within the developing embryo, they have to be constantly exchanging these signals back and forth, and then robustly and stringently establishing their regional identities relative to each other, so that the brain forms in the right place, the gut, the musculature, etc. Remarkably, these signals that are used to coordinate developmental events are highly conserved across the animal kingdom. For instance, a certain signal called fibroblast growth factor is used to instruct heart cell identity across many different kinds of animals, from a fly to a sea squirt to mammals like mice and ourselves. Additionally, when you have mutations that cause errors in these signaling pathways, that can lead to inappropriate cell behaviors. Cells can divide when they're not supposed to divide, or move outside from a tumor and metastasize and cause cancerous types of cell behaviors. And therefore, the scientific community is really committed to understanding cellular communication. And in particular, how do cells listen to these signals around them? How do they process or integrate that information? And how do they generate the appropriate response based on that complex set of conversations that are going on around them. So we study these questions in Siona intestinale. So I'm going to take a minute to tell you about this organism. And I'll first show you an adult Siona. These are invertebrate filter-feeding marine organisms. They're stuck to the substrate for their entire lives. And they are part of a larger group of animals called tunicates, which include both solitary forms, like the Siona that you saw, as well as these very fascinating colonial forms where their bodies are interlinked into colonies. They also include some free-swimming forms that can form interlinked chains of bioluminescent organisms that are quite beautiful and fascinating. And some of these chains can reach very extreme sizes, as exemplified by Pyrostrema, which can grow up to 30 meters in length. So these are really fascinating and exotic creatures, but why would you want to use this somewhat strange set of creatures to study fundamental questions about development? The primary answer is that the embryos develop in a very simple manner. In particular, they have very low cell numbers. So we can follow the fate and the decisions that are being made by every cell in that developing organism. Additionally, despite appearances, these organisms are actually our close cousins. They're part of our own chordate phyla, and they're actually the closest sister group to our own clade, the vertebrates. Okay, so now I'm going to tell you about our research as we listen into these cellular conversations that underlie the establishment of heart cell identity. So I start by telling you that the Siona heart can be traced back to 4 specific cells in the embryo that we call the founder cells, and that we can label with this fluorescent protein. Each of these founder cells divides in a highly asymmetric fashion to produce a smaller daughter cell and a larger daughter cell. And the smaller daughter cells will move into the head, as you see in this later stage embryo, and these 2 cells will form the heart. Their larger sisters remain in the tail and form tail musculature. We know that this identity of these cells is established by FGF signaling. It's very simply represented here. There are cells around them that are secreting FGF. Interestingly, both cells are exposed to that signal, but only the smaller cell responds and becomes a heart progenitor. We know this by some very simple perturbations that we've conducted. For instance, if we prevent this communication from happening by blocking FGF signaling, then the animal's not able to produce a heart. That's shown here. So here's a juvenile. This is a young Siona that now has a beating heart. This is a very simple single compartment organ. When we block that FGF signal, we see a juvenile that develops quite well and has all the other structures, but it lacks any heart tissue. That would be right here. Conversely, we can enhance that communication and basically trick those cells into thinking they've seen FGF, so that all of the cells become heart cells. And now we're going to get extra heart tissue. So here once again is the control. We have the single compartment heart right here below the stomach. And here we have a juvenile in which we've made extra heart tissue. You can see the heart is somewhat larger. You can see it's having trouble beating, but then it kind of regains its composure and starts to pump some blood. The reason this heart was having some difficulties is because there's actually now 2 compartments in this heart. There's a large compartment here and a smaller compartment here, and they're able to coordinate their beats so that they can actually efficiently pump blood. So this is a really unexpected and exciting discovery, because what we saw is that just by making more heart cells, we not only got a larger organ, but we also got a qualitative change in the type of organ, moving from a single compartment heart to a dual compartment heart. Okay. So I'm now going to move into our current research and tell you about what some of my students have been doing the last few years. So one of our big projects has to do with the idea of selective attention. As any of you know who've raised teenagers or taught teenagers, human beings are incredibly good at listening only to what they want to hear. And they can ignore everything else. And the same thing is true of embryonic cells. So as I mentioned, these 2 cells are both exposed to FGF, but only that smaller cell is responding. And we're very interested in how that's happening. Why is it that only one cell responds to the signal and becomes heart, and the other ignores the signal, like my teenagers ignore me? And this question is being addressed by my lab, and in particular by my postdoc Christina Cota, and along with a number of students, including Anna Cha, Chanel Nagone, and Nat Dreyer. Sounds like a simple question, but as you'll see, it's quite a complex answer. The first part of this answer is that these cells, before they divide, actually invade into the underlying tissue layer. And as they invade, they make these very strange-looking cuboidal invasive membranes as they surround a neighboring cell. And this allows them to actually anchor themselves to that cellular layer. As they then divide, they take the FGF receptors, which are represented here by these pink boxes, and they actually transfer those FGF receptors that are how they listen to the signal down to the anchor. So you end up with all the FGF receptors that listen to that signal localized down at that anchorage point. Now when the cell actually divides, that smaller heart cell inherits all of the receptors. So it's the only cell that can hear the FGF signal. The larger daughter cell is now deaf to the signal. It doesn't have any of the receptors needed to respond, and so it doesn't respond. This mechanism of a selective attention kind of mechanism, we think could be really important in cancer. If you break this kind of selective mechanism and you end up with more cells hearing these signals than you're supposed to have, you could have some problematic cell behaviors, overproliferation, forming tumors, etc., that underlie cancerous cell behaviors. The second project is being initiated this summer by my new student, Brighton Fett, and myself. And what we're investigating is the very simple question of how do you get two different sized cells from this division. We know from previous research that the plane of a cell division is determined by a structure called the mitotic spindle, which can be displaced towards one side of the cell, and you can get an asymmetric division with a smaller cell and a larger cell. And what we're now investigating is in the context of these Sionna heart cells, how is that spindle displaced? What are the precise molecular mechanisms that move it to cause one cell to be larger than the other, which is actually a key component of how stem cells divide in our own bodies. And the third project I'm going to briefly mention has to do with how the heart ends up on one side of the body. So you know that in human beings, our heart is always on the left side. Certain genetic mutations can lead to the heart forming on the right side or in the midline of the body. In Sionna, the heart always forms on the right. And my student, Carl Palmquist, who's a rising junior, basically established this project and has carried it through since he's a freshman. It's really his project. It's a really amazing effort. And he's recently been joined by Isabel Erickson, a freshman who's joining the lab this summer to work with Carl on this question. And what I'm showing you here is that they found a point in embryonic development when the heart cells here shown in green lie exactly on the embryonic midline. And just an hour later, they can see those heart cells all shifting to the right. And they're actually now investigating the exact signals that are causing those cells to move to the right side of the body, which is very exciting because we actually don't know why our heart cells end up moving to the left and causing our hearts to form on the left part of our own bodies. And with that, I want to once again thank all the people that contributed to this work, many of whom I mentioned. In particular, my postdoc Christina Cota has been absolutely essential in establishing my lab here at Swarthmore. And many students, including many that I've mentioned, who've really made the lab a wonderful place to work. As I think all of you know, Swarthmore students are just incredibly fun to work with. They're super engaged, brilliant, creative, and it's just a joy to have these amazing students in my lab. And with that, I'd also like to thank funding from American Heart and National Institutes of Health. With that, I thank you for your attention.